Thank you. Is this on? Yeah, okay, thank you. Thanks to the organizers for giving me the opportunity to continue a little bit, dig a little bit further into what we are doing with cyclophilins at the Julian Michel's group. And luckily for me, Antonio did all the heavy lifting in explaining why cyclophilin is important, so I'm not going to repeat that, but I will just briefly show this slide again to remind you that what we are interested is in trying to characterize structurally the minor states that are hinted by NMR studies in the hope that we can exploit those minor states for drug design purposes. The problem, of course, that we have to face is that the transitions to these minor states are occurring in very long timescales, so we didn't have when we started the project, we didn't have a clear idea how we could sample these transitions. So what we did was trying to combine all the information we had. So we started from the X-ray and some CPMG measurements published by SMSer to set up a set of accelerated MD trajectories that will help us focus the sampling into the regions that were highlighted by the NMR experiment. This led to a set of really very connected structures that were describing a rare event. And we took these structures and we set a set of equilibrium in the trajectory runs that we then analyze with Markov state modeling to create intermediate Markov states that we then resample and in an iterative cycle that we repeated until we got an MSM that was numerically consistent and that we wanted to further test. So when we wanted to, we wanted to reproduce some experimental observable, and in particular, we wanted to reproduce the CPMG measurements, or the relaxation dispersion measurements. So for those of you that are not aware, relaxation dispersion experiments, basically, they use a series of pulses of higher frequency to delete the contribution of chemical exchange to the HSQC signal. And basically, if your residue is not involved in chemical exchange, your transverse relaxation time will not change across this series of pulses. If your residue is involved in chemical exchange, it will show this kind of pattern. So Simon already talked about trying to predict this from MD yesterday. We followed a different approach. We contacted Andrew Baldwin at the University of Oxford, and we asked him to use his multi-X program where we could fit MSM rates from our trajectories and a set of chemical shifts that we compute with SIFTEX in order to predict these RX contributions. However, if we did that just with the MSM rates that we got out of the model, we discovered that we were not able to reproduce the CPMG experiments. We were just getting flat lines, which hinted that so the rates were way too fast. So what we did, we took the naive approach that we just scaled down the rates of the Markov model, and we found that if we scale it by a factor of approximately 30, we were able to nicely or qualitatively at least reproduce the CPMG spectra. That was nice, but the question remained, and I will appreciate any help that you can provide on this, is why do we need to scale down the force fields? I mean, I can take the rates, so I can take the token trash talk of the force fields here, but maybe we are also having problems with the discretization of the Markov model. It can also be that the chemical predictors are not accurate enough. So in this prediction of RX, of course, the prediction of your chemical shift is important. But we also think that maybe the way that these RX experiments are designed, where you only consider a two-state model, may be too simplistic, and maybe we should consider multiple models. So anyway, we had a look to the model with the scaled-down rates, and we could see basically that we were observing two kinds of movements. On one hand, what was the 100th loop was moving back and forth quite fast towards what we call a closed conformation, where it is collapsed on top of the 70s loop, and this will happen quite fast. On the other hand, from time to time, from the dominant state, which is very close to the X-ray, it will jump to a very lowly populated state where the 70s loop is slightly distorted, but still in place. And from there, it will go to this other state where it is really it looks like a disordered state where the 70s loop is flapping around. That was nice, but we wanted to further test the model. We compared with a set of NMR observables that were kindly given to us by Professor Vogeli. And basically, you can see that if we compare our MD ensemble, the performance of our MD ensemble against the performance of two NMR sets that were available in the PDB, we perform in a reasonably well, or at least equally, to these other two NMR ensembles in both the NOSI experiments, the coupling constants, or the residual dipolar couplings, which is nice. But if you actually go and look at the ensembles, they look quite different. So we thought that maybe there would be some degeneration of these observables, and that they are not able to completely discriminate the structures that are clearly different when you go and look at them. So we wanted to try to derive a model-based hypothesis that will help us increase our confidence in the model. So we went and looked at the Markov model and thought, let's see what is controlling what seems to be controlling the 70s, the dynamics of the 70s loop. And we found that at the heart of the 70s loop is this aspartate, right? That is at the very central node of a network of hydrogen bond interactions. In the intermediate state, that network is distorted. Some of these hydrogen bonds are still there, but some of them are no longer there. And then when it is in the what we call disordered state, the aspartate is just out of the picture. So we thought, if we take this aspartate and we mutate it to alanine, we should be able to speed the whole process, and we will have a protein with accelerated dynamics. Sure enough, we did the experiment. We built the MSM of the mutant protein, and we can see that there are two main features. First one is that the dominant state is no longer one that resembles the X-ray, but one that resembles what was the intermediate state in the wild type. And also we have a speed up of the whole dynamics of the protein. As comparison, we also mutated as a sort of negative control histidine 70, which is in the vicinity of the 70s loop, but it was not supposed to be included in the controlling this network. And you can see that if you mutate histidine to alanine, as we did with the aspartate, you get a Markov model that resembles much more the wild type. So that was tantalizing and encouraged us to actually go and commission the D66 alanine cyclophilin plasmid. And in collaboration with Arun in Edinburgh and Gox in the Baldwin lab in Oxford, we expressed the protein and took some HSQCs. And you can see that there is already here in the HSQC, there are a lot of residues in the 70s loop that go towards this skew area that is hinting that the 70s loop is disordered in the mutant protein. This can also be seen here in the CSP NMR, in the CSP plot, where it's a quantification of these displacements in the HSQC. And I would like to point out also the qualitative agreement of the prediction from the MD and the actual NMR experiment in these quantities. And also with collaboration with Harris in Edinburgh, we got a crystal of both the protein, where you can see nicely the electron density of the protein, of the loop, and of the mutant, where the electron density of the protein cannot be found. And so hints, again, to a disordered state of the loop. We also wanted to prove the microsecond to millisecond dynamics. We ran the CPMG experiment for the D66 mutant, and you can see that there are some residues that are involved in chemical exchange in both the wild type and the mutant, but the pattern doesn't look very much alike. They don't resemble each other. And in fact, if we try to do the same experiment that we did with the mutant, we are not able to reproduce these minor states that are seen in the experiment. However, if you look at the correlation between the chemical shift of the excited state of the wild type and the dominant state in the mutant, they correlate quite well. So that's hinting towards that they are dissimilar. These states are somewhat similar. However, when you try to correlate the chemical shift of the mutant with either the dominant state in the wild type or the excited state in the wild type, they don't look alike. So altogether, what that hints us is, or what that is telling us, is that the mutant does interconvert to other things, but we don't know. I mean, we don't know how that looks like. And given the design of our Markov model, it would be very difficult for us to take. Maybe we have to run some iterative sampling on them and try to capture these states. So just to conclude, we think that a combination of accelerated MD and Markov modeling can be used to describe protein movements that you cannot usually capture with equilibrium MD, that you can get qualitative agreement with the relaxation dispersion. One thing that we would like to discuss is, so the MSM kinetics seems to be 30 times faster than we should be, and we would like to know why. Of course, there is a temperature difference there that will account for a factor of 2, but still a factor of 15 that we need to explain. Then, we would like, so we think we have proved that simulation-based models can be used to generate testable hypotheses that then you can go and you can convince the experimentalists to try. And that's a nice thing and offers new opportunities for structural biology. And we are currently using the D66 model insights to design new cyclophilin inhibitors. However, we agreed that we are not yet in a state where we can actually come and show this. So last but not least, I would like to thank everybody involved in the project. So Julian over here, Tony, of course, which helped with the MSM, Charis, the crystals, Arun with the NMR, and Harris with a lot of IPCs. Also the European Commission for the funding, of course the collaborators, and the UK supercomputer structure for computational resources. And you for your attention. Thank you very much.
So as we're running a bit behind schedule, is there a burning question? Otherwise we can carry on the conversation with coffee. All right. Thank you very much. Back at 10:45.